Good morning and welcome to Lucas's super awesome presentation, hosted by yours truly, Lucas Lopez. Make sure to smash that like and subscribe button before we get started. All right. For our first slide, just going to show you my figure. We had figure 4. These are basically just showing you the difference between the blood-brain barrier tight junctions within the brains of both germ-free and pathogen-free mice. Those are the two mice types experimented on during this article. And the question is there a difference between the BBB, blood-brain barrier tight junctions, and germ-free and pathogen-free mice? Based off of this figure, which I will explain more in depth soon, yes, there is. If you just look closely, figures A through F, these ones over here, they are comparing the expression of a bunch of proteins like ZO1, occludin, and claudin-5 in the different parts of the brain, the frontal cortex, the striatum, and the hippocampus. These are proteins that, when expressed, tighten the junctions. And then, yeah, Western blot was used for A through F, and then I'll explain a bit later for the other ones. As you can see there, we do definitely have some stuff going on. So Western blot, basically what it is, like I said, it was used for A through F, and it detects and analyzes certain proteins. The proteins analyzed here were the ZO1, occludin, and claudin-5. And so a control was used for this Western blot experiment. That is the GADPH, which I can highlight here for you. The highlighter is not working, but that is okay. Basically, the one at the bottom of A, B, and C, that is a control. It is a protein that is going to be expressed guaranteed in both the germ-free and the pathogen-free mice, which is what we were testing on, kind of just to show what the test can do. And so basically, in order to do a Western blot, if you look to your right, the proteins are separated, they're transferred onto filter paper, they are stained using antibodies and blotted onto the visual. The darker the blot, the more expression there is. D through F is just a table visualization of that, of how dense the blots are, and that's just densitometric analysis, density, all that good stuff. And then it shows that in pathogen-free mice, which are the black bars there, there's a statistically greater expression of occludin and claudin-5 in all three areas of the brain, whereas ZO1 is not statistically greater in any brain area between pathogen and germ-free mice, which is quite interesting. The greater expression of most of the proteins, though, results in a tighter junction in the pathogen-free mice, which is not good. You want, which is good. You do want a tighter junction in your BBB. And I do have a little thing right to the right here that explains how it works. Next, the next images, G and H, were just basically an immunofluorescent analysis of the cerebral motor cortexes. And immunofluorescence is just a simple stain using antibodies. If you look to your right, attached to proteins and fluorophones are added for color. And there's two different ways of doing that. And so we used endothelial cells found within the motor cortex, and we stained those for occludin antibodies, claudin-5 antibodies, and laminin antibodies. Once again, some more proteins that help out with that blood-brain barrier. And we tested these both in germ-free and pathogen-free mice, just as every experiment goes. For every experiment, these are the two things we want to compare. These images confirm that there are less antibodies in germ-free mice to connect to occludin, laminin, and claudin-5. Meaning that these tight junction proteins aren't being expressed as much in pathogen-free mice, which, like I said, they are being expressed as much in pathogen-free mice, which means that they have that tighter junction, which is exactly what you want. And then this is an electron micrograph, simple electron microscopy, looking at something really small here. We're looking at the tight junction structures of endothelial cells in the brain for each type of mouse. Germ-free is on the right, pathogen-free is on the left. This just gives you a visual of the physical difference in junctions of the blood-brain barriers. Obviously, the pathogen-free mice has a decreased number of organized tight junctions, decreased number of disorganized tight junctions, and the other ones are much more disorganized due to the lack of expression of the tight junction protein. So you can see that GF is a lot more blah, 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 whereas the pathogen-free is very straight, and that's exactly what you want, and that's exactly what we're looking for, trying to find out. And then the last image, this J, This is just a tight injunction storing. They kind of created a quantitative label to number for the intact tight junctions in the endothelial cell. So it's, let's say perfectly tight is a 3, there's patches of blurriness, blah blah blah, whatever, is 2, and totally blurred is 1. The number of intact tight junctions was significantly lower in the germ-free mice, around 1.5 compared to 2 in the tight junction PF. So like I said, it's just a visualization to get some quantitative data of all that good stuff. But basically what you can pull from all this is that the germ-free blood-brain barriers are not as tight as the pathogen-free blood-brain barriers. And so, yeah.